Nope, it's working now. Okay, that's good. Yeah, okay, that's good. Okay, so I'm just gonna go into reproduction. Yes, so today we're going over reproduction. And, you know, like some of you have said, you learned it from school back when, and such. But we're gonna go more in depth into each of the different topics today. And just so you guys know, like currently we are in the 15-week full course for MCAT content review. We started June 17th, which was last Thursday, and there are 3 lectures per week, Thursday, Friday, and Saturday. All of them are at 8:30 p.m. EST. We also have one office hour session per week on Sunday at 8:30 p.m. EST. And as of now, we only have one, but this is subject to change. And if it is to change, then we'll update it on the GroupMe, which I have here, the GroupMe and the link. So overall, you know, any updates or changes about the course in general will be posted here. And if you have any questions for any of the MCAT tutors at any time after this presentation, like during the week, you can put that there and they will try to get back to you as fast as possible. And for the purposes of the 15-week MCAT course, we're going to try to cover most of the highest yield MCAT content. So today, the learning objectives are to review the cell cycle, review the processes of mitosis and meiosis, review the male and female reproductive systems, understand the hormones involved in the menstrual cycle, review fertilization and gastrulation, and review post-fertilization effects. So as you can see today, we have quite a lot of content to go through. So usually I would say that you guys could, you know, send in or unmute and ask questions during the presentation. But for the purposes of today, if you guys have any questions, please put them in the chat and the other MCAT tutors will try to respond to you as soon as possible, or keep your questions to the end of the presentation and then we can have like a conversation going at the end, just so that we can get through this content and hopefully finish before like the 10:30 mark. Okay, so the first part, as I had mentioned, we are going through the cell cycle. So, you know, last Friday, I discussed the cell, but not really how cells replicate and divide. And the cell cycle is a process in which cells continuously go through a process of growth and division. As more and more cells grow in different parts of the body for different purposes, it's really important to understand this for the MCAT and for understanding how the cell works. There are several phases to the cycle, which I listed here in the bullet points. The first is G1, or the first gap phase. And in this phase, the cell utilizes nutrients and grows larger. It copies organelles and creates building blocks to prepare for the next step. So as you can see here in the diagram, we're starting at the right side here, G1, and then we keep going to here would be the S phase. So the next phase, S phase, is where DNA synthesis takes place, and the amount of DNA is doubled in the cell. And for the purposes of this presentation, I won't go in depth about how DNA replication occurs because we will be going through the process of DNA replication more in depth in a future biology lecture. But for now, we just know that the DNA has doubled in the S phase, right? And after the S phase is the G2 phase, or the second gap phase, in which the cell continues to grow similar to G1, but is preparing for the next phase. So both the gap phases also have checkpoints prior to going to the next step. But usually the G2 checkpoint is more important, especially because it regulates, you know, cell duplication and division. And having unregulated duplication and division can lead to cancer. And so saying the duplication and division, we then go through the next phase, which is the M phase or mitosis phase, in which the cell divides and undergoes cytokinesis. We will go more in depth into, mitosis and also meiosis in the next slides, but for now, it returns the cells to the original size and contents as it was previously in the G1 phase. And this cycle, the cell cycle, repeats continuously unless the cell goes into the G0 phase, which you can see it kind of like pops up on the side over here. But the G0 phase is the resting phase where cells maintain their size and perform its normal functions usually. And also something to note, you see how there's like a smaller circle and then a larger ring. So the smaller ring, I went through each of those phases, but the larger ring has an I, which represents interphase, and that's where G1 or G0, S and G2 phases are like making up that portion. And then the M is still an M phase for mitosis. So now, speaking of mitosis, we're going into mitosis. So in mitosis, the first step is prophase, when the genome becomes visible after condensing into chromosomes, the nucleus disappears, the spindle and kinetochore fibers appear, and the central pairs begin to move to the opposite sides of the cell. As you can see, I have the diagram here of like the process of mitosis, not necessarily in the human cells, but this is in just an example with 2 chromosomes here. And so after prophase, which I just was talking about, the next step is metaphase, in which the chromosomes kind of line up at the center of the cell, which forms this metaphase plate. And they do so because the kinetochore of each sister chromatid is attached to the microtubule complex spindle fibers that are attached to, the opposite sides of the cell. And then after metaphase, we go into anaphase, which is when the spindle fibers shorten, as you can see, compared to the previous one, but they shorten and the sister chromatids begin to be pulled apart, and you begin to see a cleavage furrow. It's starting to like have indentation. And then finally, in telophase, a nuclear membrane forms around the chromosomes, and then the chromosomes decondense and the nucleolus becomes visible. Yep, okay. I also just wanted to mention that as you can see here, there's the 2N. The cell starts off as diploid in mitosis and then also ends as a 2N, a diploid cell in mitosis. And the daughter cells in mitosis are, exactly identical as the starting ones. So next, we're going to go into another process of division, which is meiosis. And you can see I have like a mini diagram here as well. But meiosis is a little more complicated. It's slightly different, but I would say several of the phases occur in a similar manner, not exactly identical, but I'll go into, each of the phases here. So prophase 1 is similar to mitosis because there are condensed chromosomes, the nucleolus disappears, spindle and kinetochore fibers appear, and the central pairs begin to move to the opposite sides of the cell. However, the difference is that homologous chromosomes, well, the first thing is that there are homologous chromosomes in meiosis, but that they also pair with each other during synapsis, right? And then after following prophase 1, we have metaphase 1, which also looks similar to metaphase of mitosis, except that the sister chromatids, as you can see in this diagram, they're aligned on top of the metaphase plate instead of like the tetrads being directly like on like they're aligned on top as opposed to being on that line, except also in the other diagram, it was vertically. And in meiosis here, the first one is like horizontally for metaphase 1. And then also, so after this phase would be anaphase 1, when homologous chromosomes separate from each other while the sister chromatids are still remaining together here. And then afterwards is telophase 1, where the cells are divided here and are considered, by this point, they're considered haploid. So the haploid number, oops, sorry. I'm just gonna, okay. Let me just double check. But okay, let me just go through this quickly. So yes, telophase, we're in telophase, and by this point, the haploid number refers to, there is already a reduction, and N, or the haploid number, refers to the total number of chromosomes found in a gamete. So a sperm or egg cell, for example. And then after telophase 1, so after you have this meiosis 1 portion, we go into meiosis 2. And this is the second cell division of meiosis. That this process occurs more like the process of mitosis, except that they end up with a haploid number of chromosomes instead. As you can see in the diagram, it happens pretty similar. The chromosomes are lining up at the equator, similar to metaphase for mitosis, and they're kind of being separated as well, moving to the different sides. The only difference is that you're going to end up here for meiosis with 4 haploid cells. Let me just see. Okay, I see there's a question. Does do the MCAT give us the pictures and ask us which phase? So sometimes they will, sometimes, well, I actually will be giving an example question later today in the next few slides of like they will sometimes show a diagram and kind of ask you guys to identify, what type of cell division, so mitosis or meiosis, and then what phase. But that would be like a discrete question for most cases. Yep. And so I'm going to go into the next slide. So, right, we just talked about mitosis and meiosis, but what's the difference between the two? What are the main differences between the two is what I kind of want to talk about now. But if you guys have any ideas of the differences right now, please feel free to put it in the chat. Any main differences you remember between the two? Recombination, yes, in meiosis, right? And one is sexual and one is not. Bisexual, I guess you mean, bisexual, I guess what was the, if you could explain a little more by that. One produces an exact replication and another is for genetic variation. Very good, guys. Great responses. Okay, so I'm going to go into the next slide just for time's sake. And so here we have mitosis versus meiosis. So I gave kind of an example here in the diagram. The top portion is mitosis, the bottom portion is meiosis. And because I feel like the MCAT likes to test this as well, like making you guys recognize the differences, I just wanted to drive home these main differences. And so one main one is that cell division occurs once in mitosis and twice in meiosis. If you remember, like there's the meiosis 1 and meiosis 2 part. And mitosis produces 2 identical diploid daughter cells, while meiosis produces 4 non-identical haploid daughter cells. You can see here, the top and then the bottom. And then also, as many of you have mentioned, the genetic material is identical in mitosis. Well, that is not the case in meiosis, because in meiosis, there's processes like crossing over. And finally, I wanted to mention that in meiosis, there is also a halved amount of genetic material from because we turn from a diploid into a haploid. And also mitosis occurs in somatic cells, while meiosis occurs in reproductive cells or gametes.
What do you mean by somatic?
Somatic, they're like body cells. So actually, I'll scratch that. Somatic cells are basically any cells except for the gametes. Yeah, skin cells are considered somatic cells, just any of them as opposed to the gametes. So gametes, we know sperm and egg. Those are the main, those are the only ones that do meiosis. And then the rest of the body cells perform mitosis. Okay, and so because of that, we're gonna go into some questions so far, just to test you guys on the material that you've learned. So I'm going to put up a poll and give you a minute. So just hold on for the poll, but think about the question for now. Okay, poll is up. Yep, and like David said, take an educated guess if you're not sure of the answer. I'm gonna give like 15 more seconds if you guys can get your last answers in, and then we're gonna go into the next one because we also have a few more questions. Okay, so that was the end. I'm gonna share the results now. So I can see here that we have 79% saying A, 7% saying B, and 14% saying C. So, well, I am proud of you guys because you can see here, the correct answer is A. Yeah, great job, guys. The correct answer is A. Yeah, so the question is, a student is examining a whitefish blastula cell under a microscope. Based on his observations, the student proposes that the whitefish blastula cell is an interphase. What observation would support the student's claim? So just looking at the question, we are trying to determine the difference between if a cell is an interphase as opposed to, another phase. And if we're looking at that larger ring that I showed earlier before, we know that in those instances, the only thing that's included is interphase and mitosis. So from this, we would kind of know that anything that's related to mitosis at all would probably not be the correct answer. So as we can see here, I'm going to go for B first. The mitotic spindle is fully assembled with its poles at opposite ends of the cell. And so here we already see the question saying the mitotic spindle. And so because this process is involved in mitosis, then we know that it's mitosis as opposed to interphase. For C, sister chromatids have separated and are moving towards opposite poles of the cell, which is also usually like in the anaphase portion of mitosis is when the sister chromatids would do that. And then finally, the cleavage furrow has formed on the cell surface, and a cleavage furrow is usually in that between anaphase to telophase when the cells are already about to separate and create new cell membranes so that they can be separated into the two daughter cells. And that's why A is the correct answer. So the genetic content of the cell exists as dispersed chromatin, because you know that when you're going for prophase of mitosis, the dispersed chromatin right before it's going to start, going through the process, it kind of condenses so that that genetic material can be easily, separated throughout the processes of mitosis. Oh, go over why B is wrong again. Okay, so B says the mitotic spindle is fully assembled with its poles at opposite ends of the cell. Now, in order for the mitotic spindle to be fully assembled, that means that the cell is preparing itself already for, or that the cell is already undergoing the first phase of mitosis, so prophase, right? And we're looking for a whitefish blastula cell that is an interphase. And so interphase is any of the other phases. So G1 or G0, S and G2, which are, the phases aside from mitosis. So any answer choice related to mitosis for the purposes of this question would be incorrect. Does that help with explanation for why B is wrong? Okay, great. Of course, yes. And now we're going to go into another question. So I also wanted to incorporate graphs a little bit in this one. I feel like it's on more, it's based on what we've just learned with the cell cycle, but I just wanted to show you how the MCAT could also contribute or integrate using graphs into the content that we've just learned. And if the text is really small for you guys at the top, it says the following graph shows how cellular DNA content changes over the course of a typical mitotic cell cycle in rapidly proliferating human cells. And I'm gonna also put up a poll in a bit, and I'm gonna give you guys a minute for that. Okay, I'm gonna give you guys like 15 more seconds if you can get your last answers in. If you don't know right now, just make an educated guess. Yeah. Okay, closing poll now. Okay, I'm sharing the results here, if you guys can see it, but I'm gonna say, so 87% of you guys said B, and 7% of you guys said A, and 7% of you guys also said C. So I'm very proud of you guys because you couldn't tell with my facial reaction. The correct answer is B. So, phase B, because the amount of cellular DNA is doubled during this phase. And so, the question saying, based on the graph, which of the following labeled phases corresponds to S phase of the cell cycle? We see here that the y-axis is DNA content in picograms, and then the x-axis is time in hours. What we definitely know about the S phase is that DNA is doubled during that time, right? So if we see on the y-axis that, can I go over the G1 phase again? Sure, yeah, I'm gonna go over it right after I just quickly go over this explanation. Okay, so, right, so we see here that at the starting time, there is 6 picograms of DNA, and when the cell is going through S phase, we know that that value is going to double at some point. And so it's only phase B here that's showing that change from the 6 to 12. So after S phase, it completely doubles to 12. And so that's why B is correct. For those who chose A, said phase A because it's the longest phase of the cell cycle. Actually, S phase is arguably relatively short compared to the G1 and G2 phases, but also, as you guys know, like the main purpose of S phase is for DNA duplication. And so even though, for phase A, it's constantly staying at 6 picograms of DNA, which is why choice A is incorrect. And then for C, phase C, because it occurs immediately after the amount of cellular DNA is doubled, actually, after the DNA is doubled, that would go into the G2 phase instead. And then for D, phase D, because the amount of cellular DNA is halved during this phase, in the S phase, the cellular DNA is doubled, not halved, just to go through that. And then it mentioned going over G1. So I'm here. So for G1, mainly the purpose of G1 is the first gap phase where the cell utilizes nutrients and grows larger. That's where it makes the building blocks for the next phases for, DNA replication and more. And, the cell is basically just growing and preparing itself as time goes on during this phase. Is that good?
Hi, what is it? I'm just confused, not confused, but what does it mean to copy organelles? Like, what is that?
Oh, what it means to copy organelles?
Yes.
So so within a cell, sometimes there are multiple, lysosomes or multiple endoplasmic reticulum kind of around the nucleus. So they're preparing itself to become this mature cell.
Okay. I think I kind of got that confused with that and the S phase, like copies, kind of doubles.
Yes. Yeah. So let's focus on S phase being completely just for DNA. That's all really you guys need to know for the purposes of S phase. It doubles the DNA content. Everything else regarding the organelles, nutrients, anything that the cell needs, that would be found in the G1 or G2 phases. And then for, cellular division, that would be mitosis.
Thanks.
So thank you for asking a question. about that. Okay. Yeah, and if anything's, confusing to you guys, because at least for me, I know reproduction was a little bit it was a little bit complicated, but yeah, feel free to put in the chat. Tutors are helping to answer questions. And also, if we're in the question portion of the slides, I can also answer them. I see. So you said after DNA is doubled, it goes into which phase? So after DNA is doubled, it goes into the G2 phase, as you can see in this diagram. And also Chris answered that question too. I just realized. Okay. But yeah.
I have a question with that quickly though. I know
Mhm.
it's probably premature and you'll probably go over it in later lectures, but it's just such a smooth cycle. Does it always just go as planned? M to G1 to S, G2? Does it, are there checkpoints? Do we need to get into checkpoints and all of that? Or that's later?
So for the purposes of this lesson today, just because of the other content that we have to get, I will not be going like into depth on the checkpoints, but I did mention the checkpoints earlier. So there are usually checkpoints at G1 and G2. I feel like especially the G2 checkpoints are very important because that's, regulating cellular division. And once that kind of goes awry, it can lead to more problems.
And the last thing, you had said that whatever the cell needs, it gets it in G1 and G2. Are you talking about
usually?
What would the cell need? You said whatever the cell need, it gets it in G1 and G2. Are you talking about?
Like nutrients.
Oh, food.
Nutrients to continue to grow larger, build its building blocks.
Makes sense. Yeah. Okay, that makes sense. Thank you.
Yep. And also regarding the G1 and G2 checkpoints, really quick, you guys can probably really easily search that up as well. They have usually main checkpoints associated with each of them. One is like P53, I think, is for G1, and others for G2. But usually, if they are to ask a question about checkpoints, they would probably provide more context than just saying, if P53, just went crazy or something, what would happen to the cell? They would probably have more context as to, either the checkpoint, or they'll probably have it in a passage and be talking about the cell cycle in the passage. Okay, so we're going to go into actually I had more questions, but yeah, so we're going to go into this just to go over, meiosis and mitosis that I went over. I'm going to put up the poll. I'm going to give you guys a minute. And the polls are all anonymous, by the way. Okay, so 15 more seconds, guys. Okay, I'm ending poll now. Okay, so I'm going to share the results. So we have 64% saying B, 7% saying C, and 29% saying D. So I will say the majority of you guys got it correct, so it is B. And I could tell, I guess there was confusion with D and C. So the question is asking, right, which of the following occurs specifically in meiosis 2? So let's go through the answer choices. So A, the doubling of genetic information. A is incorrect because the doubling of genetic information happens before either mitosis or meiosis. And so, you know, that would happen in S phase, which is not related to meiosis 2. For choice C, the formation of 2 diploid daughter cells. If you guys remembered the distinctions between mitosis and meiosis, mitosis forms 2 diploid daughter cells, while meiosis produces 4 haploid cells, haploid daughter cells. And then for D, I can understand here, so crossing over of homologous pairs. I know I mentioned that homologous pairs are associated with meiosis, but they are specifically associated with only the meiosis 1 portion of meiosis. As you guys remember, the homologous pairs are the ones that are pairing earlier on, but once they finish meiosis 1, right? Once they finish meiosis 1, they get, you know, they get further pulled apart, and then the focus is more on the separation of sister chromatids, which leads into why B is the correct answer. Crossing over happens during synapsis, which is during prophase 1 of meiosis 1. And then after, you know, once you get the reduction from the original diploid cell at the end of meiosis 1, that's when you would be going through the rest of the process to get to the 4 haploid cells. And as Chris, mentioned, make sure you understand the difference between sister chromatids and homologous chromosomes, in which, you know, sister chromatids are identical pairs, homologous chromosomes are non-identical. Put it in the chat. Okay, and so this next question is one in which I was kind of hinting to earlier in the lecture. So this is something that you might see on the MCAT to like apply your knowledge of both of these types of cell replication or division for on a diagram aspect. But I'm going to put up a poll, and I'm also going to give you guys a minute again. And this will be the last question that I have before we go into more content. But make sure to keep this content in your mind as well, because it helps for the rest of the content. Okay, so I'm going to give you guys like 10 more seconds. Get your last answers in. Okay, I'm going to end polling now. Okay, I'm going to share results with everyone. So here we have 64% saying A, 21% saying B, and 14% saying C. Yep. So, I mean, I'm still proud of you guys because the majority of you guys got it correct. So the answer is A. And I guess I'll go into each of the choices. So we see the question asks, what stage of cell division does this diagram represent? And when we're looking at this diagram, right, we're seeing that the first distinction, these are homologous chromosomes as opposed to sister chromatids. So that's already one hint. And then second, that, you know, they're being pulled apart as opposed to being on, like, they're being pulled apart as opposed to being on a metaphase plate or anything like that. So when we're going into the questions, we see that the choices are between mitosis and meiosis one of metaphase or anaphase. So for the purposes of C and D, we know that the stage of cell division is not currently in metaphase, just because, you know, they're not directly lined either vertically or horizontally, the homologous chromosomes or the chromatids. But yes, so that's how we eliminate C and D. Now, if we go into A and B, so B says mitosis anaphase. Now, mitosis anaphase would look a little different as opposed to what we have here, which is meiosis 1 anaphase. So mitosis anaphase would, if you guys remember from the previous diagram, actually, maybe I'm just going to refer to it so that it's easier to visualize. This one is so we see here in mitosis anaphase where, you know, we only have these sister chromatids here, as opposed to like the full homologous chromosome. And right, nice tip from David. Metaphase, remember middle. That's great. But yeah, these two processes are something that you guys should definitely know for the MCAT. And yeah, if you guys have any other questions, feel free to ask now in the chat.
Could you repeat what was the distinguishing feature of mitosis anaphase?
Mitosis anaphase, right? Yeah. So here, instead of what you are seeing here, you would see like half of it. So it would just be the sister chromatids that are being pulled apart, as opposed to the homologous chromosomes being pulled apart. So the sister chromatids would just be like one of these. If you can see where my mouse is circling right now. Does that help?
Yes. Yes, I can see it now. Thank you.
Yes. Okay, great. Thank you. Okay. Yep. So I guess in that case, I will just keep moving forward. So now we're kind of going into the sector of going through the male reproductive system anatomy. And so first, you know, we're going through, well, I just want to start off with saying that some of the more important parts of the male reproductive system anatomy that you guys should know are the seminiferous tubules, which are the site of spermatogenesis. Oh, nice, an anaphase away. You guys, yes, that is great. Yeah, so the seminiferous tubules are the site of spermatogenesis. Spermatogenesis is the process of making mature spermatozoa, which starts at puberty and occurs throughout the adult life. It usually requires sustentacular, or it's more commonly known as Sertoli cells, in the walls of the tubules to nurture that developing sperm physically and chemically. And then, you know, between the seminiferous tubules, we have the interstitial or Leydig cells that are important for creating androgens or testosterone. which are important for the maintenance of the male external genitalia and also secondary characteristics. And then I guess a very commonly like tidbit that's tested on the MCAT is the ejaculatory pathway. It's one that you'd probably need to know. We go from the seminiferous tubules to the epididymis to the vas deferens, then to the ejaculatory duct and urethra, and finally the penis. And yeah, exactly, Opal, 7-Up. 7-Up is the mnemonic which I've definitely used so much before during my time memorizing this. It's definitely great. Probably should have bolded each of these, but yeah, so as you know, Chris mentioned in the chat, that's what each of the, so S for seminiferous tubules, E for epididymis, V for vas deferens, E for ejaculatory duct. I kind of just think of N as nothing, and then U as urethra, and P as penis. So yeah, that was a very helpful mnemonic that I used before, and I hope that you guys can use it too. So now, after the male reproductive system anatomy, we're going into the female reproductive system anatomy. So first, so oogenesis, so as opposed to, you know, spermatogenesis, oogenesis is the process of making ova, which begins before birth, and oogonia are mainly arrested in prophase 1. There are about 7 million primary oocytes at this point, but you know, this number changes to 2 million at birth, and then it also further changes to about like 400,000 at puberty, and then only 400 oocytes are ever actually ovulated. So if you're thinking of that, that's like very little from the overall number that happens at puberty. But so if we continue into, you know, meiotic division produces a large secondary oocyte and then a small polar body. And then this oocyte is what remains in prophase 1 until a sperm is fused with it. And then after the sperm fuses with it, that's when it can continue the rest of meiosis. But before that, it's just stuck in prophase 1. And then I also wanted to talk about granulosa cells, which are important because they assist in the primary oocyte maturation. Also, the oocyte plus the granulosa is considered a follicle, just for terminology that you guys can know. And then also, you know, kind of similar with the male anatomy, I'm also going through the direction of, in this case, ova in the female reproductive tract, which goes from the ovaries, then to the fallopian tube, then to the uterus, then to the cervix, and then finally to the vaginal canal. Unfortunately, I don't have a mnemonic for this one, so you guys will kind of have to memorize that one just as is. But as Mariah asks, what's a small polar body? So a small polar body, it's kind of just like, so when oogenesis occurs, the secondary oocyte is disproportionately usually larger than the other small polar body that's being, you know, produced from meiosis 1. The small polar body, it really doesn't have that much inside of it. From what I remember, it's just like cytoplasm and nothing that really helps with any DNA. Yeah, I think it mainly just consists of cytoplasm, but it doesn't allow, like, so this small polar body isn't further, it's not going through meiosis 2 anymore. It kind of just arrests at that point, and it's not really used for the rest of oogenesis. Yeah. Yeah, okay. So now we're gonna go into next portion, the menstrual cycle. Yep. So this is I feel like this can be a complicated process as well, especially with the different types of phases and the ovarian cycle versus the uterine cycle. But I personally really like these graphs that I have to the right, which kind of, lists out every single thing that's happening with the hormone levels, with the phases. It shows the diagrams of the endometrium lining and also with ovulation. So you guys should look over this diagram more if you have the chance. But yeah, so the menstrual cycle, many of you may know, but it's on an average of a 28-day cycle. It's when the primary oocytes that are destined to be ovulated gets developed in the ovaries. And then meiosis 1 is completed, and then it gets and then the secondary oocyte gets ejected with the hope of being fertilized. So as you can see in the chart to the right, it kind of shows, as I mentioned, the levels of hormones. And the main phases that I was talking about in the bullet points to the left are those of the ovarian cycle, which I feel like they tend to be tested more on the MCAT than the uterine cycle, which can be seen here in the last portion of the chart to the right, the uterine cycle with the endometrium lining. But that's why I kind of put the main takeaways for the different hormone levels for each of the phases here. Yeah. So in terms of the hormones, the anterior pituitary and the hypothalamus play a really large role in hormone secretions, but I will not be talking as much about like all of the hormone secretions from the anterior pituitary and the hypothalamus in this lesson, because we will actually have a future endocrine system MCAT lesson, and that one will go over, the signaling between for hormone release more in depth there. So for today, I'm just going to talk about the hormones that are related to the gonadotropin releasing hormones from the hypothalamus, which stimulates the release of follicle stimulating hormone, FSH, and luteinizing hormone, LH, from the anterior pituitary. So I thought something to know is that in the ovulation phase, so that's when, that's when the secondary oocyte is released. And in the luteal phase, that's when the corpus luteum is getting formed. So I show here in the follicular phase, in the ovulation phase, and luteal phase. But in the follicular phase, I also wanted to mention that so the follicle matures and secretes estrogen and also usually FSH. And well, FSH is increasing. Luteinizing hormone is still kind of stable at this point. And progesterone is low and estrogen is kind of low for a bit, and then it increases after a little bit of time. Right. And then for the ovulation phase here, you can see that many of these hormones are at peak. So FSH is, really increasing here. LH is spiking here. Estrogen is increasing. But the only kind of different hormone in this particular area is that progesterone ends up being very low. And then when you go into the luteal phase, FSH kind of comes back down. LH is kind of a little stagnant still. It's not that different from the previous one. It's just, equal here. And then estrogen it goes down, but then it increases here. And then progesterone also increases a lot afterwards. Question here. Are FSH and progesterone inverse? FSH and progesterone. Hmm. And oh, I see. Estrogen, sorry, FSH and progesterone, right? That was the question.
Yeah,
But I don't.
is it one inverse? So FSH would be higher than progesterone is higher? Because I know if I'm wrong, not wrong, but correct me if I'm wrong, progesterone is like contractions and FSH is kind of like stabilizes. So one is always higher than the other. They're inversely. Is that a good way to remember that?
I usually think of it as FSH is what stimulates estrogen, and then estrogen later stimulates LH surges. But then like estrogen and progesterone are still kind of like similar levels in the luteal phase. So in that case, like I wouldn't exactly consider them to be like inversely related, FSH and progesterone. I think it's a matter of yeah, I wouldn't automatically think that they're inversely related. I would more likely like to go through the whole process and see how like the interactions are there. I wouldn't, yeah, I wouldn't automatically say that right away, if that makes sense. Sorry. But yeah, I guess to keep going on about this. So, okay, so something to know, I guess, is that if you wanted to also know about the uterine cycle, the main phases are menses or menstruation, where the corpus luteum degenerates and estrogen and progesterone levels drop sharply. And then the next here is the proliferative phase. And then after that is the secretory phase, where estrogen induces the proliferation of a new endometrium with the continued development of this new endometrium. As you can see throughout this process, it's just continuing to develop. And yeah, as I touched on earlier, you know, FSH does stimulate estrogen. Here you can kind of see in this portion. And then this high amount of estrogen is what leads to the LH surge. So you can kind of determine the relationship between estrogen and LH. You know, those estrogen will lead to LH. And then when there is this high surge of, you know, FSH and LH, that's when the ovum is released. And then the corpus luteum is the one that's producing the progesterone. And so, yeah, and then estrogen and progesterone are continued to be produced by the ovaries as time goes on here. So I guess to go back to that question. So when FH like increases, that's when, you know, the ovum is released, and then that's how the corpus luteum produces progesterone. So for it to, so FSH, the spike in FSH is what leads to, you know, the higher progesterone levels later on. So in that case, it would kind of seem more like it leads to more progesterone happening as opposed to being inversely related with it. Yep. Hopefully that answered the question. Yeah. If you guys have any more questions, feel free to put it in the chat. And also we can have a discussion at the end of this PowerPoint because we still have a few more slides to get through. So the menstrual cycle, definitely, you know the interactions between the hormones. for this particular slide, I kind of like listed all of the hormones, you know, under each bullet point, but I think it would also be helpful if you guys created a chart. At least that's what I did. I kind of created a chart like FSH, LH, estrogen, progesterone, and then I put each of the phases on the left side. So it's just easier to visualize, and you can look back at that when you just want to review the levels. But yeah, so the next portion that we're going to talk about is fertilization and implantation. So fertilization, as many of you guys may know, is the fusion of a spermatozoan with the secondary oocyte. The sperm uses the acrosome reaction, so that's at the head portion of the sperm, in order to penetrate the corona radiata, because it has in that sperm head there are hydrolytic enzymes that are released by exocytosis, and then that's how they're able to, you know, penetrate that area. Next, I also wanted to point out some other few terms. I feel like these are not tested as much on the MCAT, but it's just something to mention here for the purposes of going over fertilization. But actin and bindin, so both of them are used to elongate towards the, so actin is used to elongate towards the zona pellucida, and then bindin is used to bind to the receptors in the zona pellucida. And then finally, when the sperm and egg plasma membranes fuse, like when you're going more to the right, the sperm nucleus enters the secondary oocyte, and then that's how the oocyte can complete meiosis too. And that's then how the ovum and spermatozoa fuse to make the zygote. So yeah. And then after this, I'm going into a quick question about, you know, what you guys have learned so far. So I'm going to put up a poll right now. I'm going to give you guys like a minute, and we'll see. Hopefully you guys remember the mnemonic. 7-Up, hint, hint. And I know some terms here were not mentioned in the PowerPoint, but if you go through the pathway, you'll see where some of the other answer choices would be made wrong, even if you were to try to use other ones. Yeah. Okay, I'm gonna give you guys like 15 more seconds. Just take an educated guess. And I feel like it's helpful sometimes when you look at the first term and the end term, and like go that way. I'm gonna give you guys like 15 more seconds. Okay, I'm gonna end polling. Okay, so we have 93% saying C and 7% saying D. So, you know, you guys, majority of you guys got it correct. I'm so proud. But yeah, so it's asking what is the correct trajectory of sperm during ejaculation. And as we know, with the 7-Up mnemonic, we can kind of eliminate the other choices just by this. So 7-up, we know the seminal vesicles and then the epididymis and then the vas deferens. And so we already see here, at least for choice D, we see that vas deferens is here and then the ejaculatory duct and then back to ampulla of vas deferens, right? And both of these portions are from the vas deferens. And if we're going by 7-up, the ejaculatory duct should be happening after any portion of the vas deferens, which is why D would be the incorrect answer in this case. And then for A, we know that the epididymis occurs before the ejaculatory duct, so that's already for A, the first two terms are already what eliminates the answer choices. But then for B, we know that the vas deferens has to occur before the ejaculatory duct, so that's why after the second and third comma, we know that that's where it's wrong, and then that's why for C, it definitely goes through the right process, although it doesn't directly mention seminal vesicles. We go through the epididymis, the vas deferens, the ampulla of the vas deferens, then finally ejaculatory duct and urethra. So yep. If you guys have any questions about this one in particular, feel free to unmute right now. If not, we're gonna go into the next question. And I'm just gonna put up the poll for a minute and I can check the chat while this is happening. Oh, where are the Sertoli cells located? Okay, just the Sertoli cells are located near the walls of the let me see if I can pull it back. So the Sertoli cells are in the walls of the seminiferous tubules. I don't have. I know. Oh, sorry. Elizabeth, if you could kindly mute, that would be great. But yeah, in this particular diagram, I don't have the seminiferous tubules. But wait, sorry. Where are the Sertoli cells located in seminiferous tubule epididymis vas deferens? Yeah, so it's located in the walls of the seminiferous tubule. Yeah, I just don't have the seminiferous tubule in this particular diagram just because unfortunately, like sometimes it's hard to find diagrams, but yeah, that's just something you guys should know, and that Sertoli cell's main function is to help nurture the developing sperm. I'm gonna go through back to the question. Oops. So yes, we are here. Right. Okay, so I gave you guys quite a bunch of time. If you guys can get your last answers in 15 seconds, that'd be great. Okay, I'm gonna close the poll now just so we can get to the next one. But okay, so I'm gonna share the results. So here we have 45% saying B, 36% saying C, 9% saying A, and 9% saying D. Okay, so oh wait, did I stop sharing results? Okay. Yep. Okay. So here, like the majority of you guys got it correct. It is B. So, but I did notice that there was quite a larger distribution here. So we're gonna, if you guys have any questions after this explanation, feel free to unmute and talk, or also mention it in the chat. But so the question asks, which hormonal change is linked with the menses period of the uterine cycle? And if we remember back from the chart, in the menses period, that's when the estrogen and progesterone levels like quickly drop in the beginning, at least from the luteal phase back to the menses period. So, oh, that's definitely why B is correct. But so choice A, increased secretion of progesterone by the corpus luteum. If you guys remember in the diagram, I guess I'm just going to go back to the diagram just so you guys can see it, what I'm talking about. But so when we go back to the diagram here, we see the menses period is in this like front portion of the follicular phase. And we see that, oh, you have not seen reproductive questions a lot. In general, like on the MCAT, or just like, I guess, in your experience? Oh, on full length. Yeah, they like to ask like the hormone questions every now and then. They'll just have like a discrete question with FSH or LH or estrogen or progesterone. I haven't seen like a full passage about like the menstrual cycle very often. Like I have not seen that, but I've definitely noticed that they do like to put like a few like discrete questions here and there. So if you guys know this like down in pat, it's easy points to get those discrete questions correct. But yeah, so as I was talking for the purposes of the question, the menses phase, so after this portion of the luteal phase, right, we go back to the beginning here, and estrogen and progesterone levels have dropped. But when the corpus luteum is formed, it's producing progesterone, but that is strictly in the luteal phase, which if you were to compare that to the uterine cycle portion, it would be the secretory phase. And then to continue on back to this one. So we have next for choice C, increased levels of follicle stimulating hormone and luteinizing hormone. And we know that the increased levels of FSH and LH happen in that ovulation phase, in that between like that day 14 period where an ovum is being released. And that is like after the menses phase, if you guys remember from that diagram. And then finally, for D, increased secretion of estrogen. The increased secretion of estrogen actually also is happening as the ovulation phase is starting to get there. So that is not quite at the menses phase. That menses phase is that beginning like portion where after the luteal phase, all of the estrogen and progesterone levels have, dropped tremendously. But yeah, that's why B is the correct answer in this case. Does anyone have questions about this particular question?
Yeah, mine is kind of indirectly related because now if the menses, during your menses, you have the decreased levels of progesterone. What about after during menopause when you're no longer menstruating? How does the FSH and LH level?
Oh, I see. So
these are the answer choices.
Okay, so menopause, the cessation of the menstrual cycle. What are the levels of FSH and LH? So A, increased FSH, decreased LH. B, decreased FSH, increased LH. C, decreased FSH, decreased LH, and D, increased FSH and increased LH. So If we can go back, I want to go back to the diagram again, just for reference. But right, we see here that from the luteal phase, which would be like after that, after the corpus luteum has been formed and has been releasing progesterone, and after it degenerates, that's when we go into the menses phase. And so instead of FSH and LH being decreased, I agree with Chris. Both of them should be increased. So I believe the answer is D. Is that correct?
I'm checking it right now. I mean, because I just assumed being that you're no longer menstruating, these hormone levels would probably, like you said, decrease. Why would it increase if you're no longer?
Oh, estrogen and progesterone would decrease.
Oh,
FSH and
oh,
LH would
that's the, okay.
that was the sorry,
Oh,
yeah.
okay, I was okay, it's estrogen and, okay, not LHFS. Okay. All right, so I guess that answer makes sense. Was passage 6?
Yeah, especially in the menstrual cycle, I feel like the hormone levels can get confusing because there are like FSH, LH, estrogen, progesterone, and they kind of, you know, go different ways throughout the process. Oh yes, okay, answer is D. It's good. Yeah, so when you're going into menopause, you then kind of go into this menses phase after the luteal phase. But I do actually have another question after that one, so we're gonna go into that right now. So this one, I'm gonna put up a poll right now. If you guys want to answer a question, I'm gonna give you guys a minute. Okay, guys, I'm gonna give you guys like 15 more seconds. And then I'm gonna close this out. So please get your answers in. Okay, I'm ending poll now. I'm going to share the results here. We have 42% saying D, 33% saying B, and 25% saying C. So, I mean, the good thing is that the majority did get it correct. Here, I'm just going to show the answer. The correct answer is D. So, but this one was also a very big split. So if you guys have any questions afterwards, yeah, feel free to unmute or put in the chat. But the question asked, during a pregnancy, hormones play a big role in maintaining pregnancy. Which of the following can potentially lead to miscarriages during pregnancy? I kind of, I guess, wanted to preemptively talk about pregnancy before we go into that portion. But if you guys remember from, I guess this might be a little bit of review / previous knowledge, but during pregnancy, we want to maintain that corpus luteum in order for implantation to be able to occur. And so then we're kind of, if we go to the previous, you remember the previous diagram, then we're kind of focusing on that luteal phase and trying to figure out how or what are the methods that we can do to maintain that corpus luteum portion and not go into the menses phase, right? And so for choice A, it's saying increasing levels of estrogen. And as you remember, in the luteal phase, both estrogen and progesterone levels are high, and they're helping to maintain that corpus luteum, which is helpful for pregnancy. So it would not lead to miscarriages if you were, well, there would be a smaller chance that increasing levels of estrogen would potentially lead to miscarriages during pregnancy. Following that, B is kind of similar, stimulation of progesterone secretory tissues. We want the progesterone levels to be high during that luteal phase and for maintaining the corpus luteum. C, decreasing the levels of oxytocin. Oxytocin, I haven't mentioned yet, but I will mention actually in a few more slides. But that's more related to the contractions later on towards the end of the pregnancy. So that's not really leading to miscarriages earlier on in pregnancy. But for D, early degeneration of the corpus luteum, if the corpus luteum has degenerated, then we're just going straight into the menses phase, as opposed to being able to, develop it for pregnancy. And as Chris said, and I will mention later on, progesterone is maintained throughout pregnancy. And in the beginning, the corpus luteum is helping to produce this progesterone. But as you reach 6 months, that's when the placenta will start producing progesterone on its own as well, to maintain these high levels throughout pregnancy. If you guys don't have any more questions, then we're just going to go into the next slide. Okay, so now we're going to go into changes during pregnancy. And so, as I was kind of mentioning before, so the menstrual cycle is the process where the oocyte does not get fertilized. But now we're kind of thinking, what happens when it does get fertilized? And in the previous question, I was talking about pregnancy. So after ovulation, the secondary oocyte is ejected, and then when the sperm fuses with, and then that has the opportunity for the sperm to fuse with the egg. But I think the other question that I have after talking about, the corpus luteum and the uterine cycle is how exactly is the endometrium also retained for the embryo? And so just to reiterate the luteal phase, during the luteal phase, FSH is decreasing, LH is kind of still stable, estrogen is increasing, and progesterone is also increasing. And then usually estrogen and progesterone levels, as I mentioned, would decrease and lead to the menstrual cycle, but particularly in pregnancy, estrogen levels increase and progesterone levels increase. And this, is maintained and inhibits luteinizing hormone release instead. But the problem here is that usually when luteinizing hormone drops, the corpus luteum also degenerates. So we kind of have a question as like, how would we still maintain the corpus luteum if we have this rapid LH drop because of the estrogen and progesterone levels? And so here's where there's a new hormone being released, which is the HCG, or the human chorionic gonadotropin. And that is released to maintain the corpus luteum, to further increase the development of the endometrium, which allows for implantation and continued growth, of the zygote. But HCG, so I show this pregnancy test here in the lower right-hand area, not the corner, but the right-hand area. But so pregnancy tests are looking for that HCG, because that definitely means that you are pregnant. So just fun fact about that, I guess. Yeah. Now we're gonna go into, so on top of, understanding the changes during pregnancy, we also want to learn about embryogenesis and development of that zygote. And, I guess embryogenesis and gastrulation, I didn't exactly want to go through like the whole process in this PowerPoint slide yet. But for the purposes of gastrulation, I really wanted to focus on like the different 3 main layers and then what they turn into, because that is also commonly tested as a discrete question on the MCAT. But yeah, so after the zygote forms, it undergoes processes of cleavage, turns into a morula, and then next into a blastocyst. And then it implants into the endometrium about a week after fertilization. So it continues to develop afterwards with the same hormone levels of estrogen and progesterone. And as I mentioned earlier, until 6 months of pregnancy. And at that 6 months is when the placenta itself can produce its amounts of estrogen and progesterone that it needs on its own. And then after implantation is when like gastrulation and the starting of the development of the embryo will occur. And so that's when the primary germ layers become distinct. And the primary germ layers are the ectoderm, the mesoderm, and the endoderm. And right, the main thing to know is, what parts of the human body are derived from each of the layers. And so in the ectoderm, that leads to the integumentary system, the epidermis, hair, nails, nose, mouth, and anal canal epithelia. Then also the central nervous system, which includes the adrenal medulla. That's also kind of like a fun fact that I think they've had like a flashcard question about that one, but I thought that was a little tricky before. And then eye lens and inner ear. And also regarding the ectoderm, there is the process of neurulation that that explains the development of the nervous system, but particularly because the reproductive system itself is already kind of a large topic today. I didn't exactly want to go over it in this lecture, but we are going to have a nervous system lecture next week. So I think that that's where, the beginning process of neurulation can be mentioned there. And so next, the mesoderm leads to the circulatory system, musculoskeletal system, and parts of the urinary and reproductive systems, which are includes the adrenal cortex and gonads, and then also the dermis, mesoderm and dermis. And then finally, there is the endoderm, which leads to the organs of the digestive system, the respiratory system, bladder, and reproductive tracts. And also here I wanted to mention, so as the cells are slowly going into each of these specific human body organs, they are slowly getting more and more specific and more determined. But they start off as totipotent, which means that they can turn into any cell of the body. And then they slowly go into pluripotent and multipotent, and then finally into their mature cells. So, commonly when we think of human stem cells, they are in the multipotent state. And then the rest of most of the other cells in our body are in the mature cell state. And pluripotent, totipotent, totipotent can turn into everything. Pluripotent is a little bit more specific in the sense that they would only be like within that particular like system or area or layer. And then multipotent, more specific, and mature cells, like exactly what the function is. And then I also included this diagram at the bottom to just visualize some of the different developments of each of the layers. So yeah. And now we're going into the next slide, which is, So the oh, so the controversy with stem cells is because you get it from embryos, not because it's so potent. I mean, some stem cells they get from embryos, yeah. By the time that they are in that embryo state, though, it's not because it's totipotent. Totipotent is like in those like beginning, blastocyst type stages. By the time you're getting into the embryo, you're already getting more specific, more into that pluripotent stage. But I guess I don't know, the controversy with stem cells. You know what? Let's go into the rest of the slides before I continue talking about that. But yes, so as I was talking about the primary germ layers, as I was talking about the primary germ layers, so I'm going to also talk about development during pregnancy. This is, I feel like, definitely not high yield as some things. I know there's just like one straight question that I've seen before with like asking what is forming during some of these trimesters. So I'm including it here, but I feel like it's definitely not the most important thing to get out of this presentation. So I'm just going to go over it briefly. So during the first trimester, that's when gastrulation, neurulation, and all major organ systems appear. And then in the second trimester, that's when organs and organ systems of the fetus continue to develop structurally and functionally. And about this time is when the fetus is about 0.6 kilograms. And then finally, in the third trimester is when there's rapid fetal growth and significant deposition of adipose tissue. And most of the organ systems are fully functional at this stage. So when there are premature babies at this like third trimester stage, since their organ systems are mostly fully functional, they should have a good ability to survive. And so now we're going into birth and lactation. So as you guys may recognize here, I have a picture of Baby Yoda. I know Baby Yoda is not a human baby, but if you guys are, do any of you guys watch The Mandalorian? Because like I'm a very big fan. Please mention in the chat if you are a fan of that show. But yes, included an image of Baby Yoda here. But talking about the hormones, so the most important hormones associated with birth is oxytocin, it was briefly mentioned in that question earlier as an answer choice. But as I mentioned, it causes the uterine contractions to increase in intensity, causing greater pressure on the cervix that produces a positive feedback loop until after birth is when it stops. So as oxytocin increases, uterine contractions increase, which also leads to oxytocin increasing more and so on. And oxytocin is actually produced by the posterior pituitary instead of the anterior pituitary. And for the different stages of birth, so for this slide, oxytocin is definitely something that you guys should know. But the first stage, dilation of cervix, the second stage is then the baby moves through the cervix and birth canal. And third stage is when there's the expulsion of the placenta. These stages, not as high yield, just something to know with this information that we already have here. But then also another important hormone to know about is prolactin, which is for milk letdown. And so usually women maintain moderate to high levels of estrogen and progesterone throughout their life, which usually inhibits prolactin release. But because after they give birth, the levels of estrogen and progesterone levels decrease even more drastically than like what you would see in the luteal phase to menses phase, which actually is what causes milk letdown from prolactin, and for prolactin to not be inhibited by them. And then we have reached the end of the slides today. So thank you guys so much for attending today. I know it was kind of long. So reminder, we have office hours, every Sunday from 8:30 to 10:30 p.m. EST. If you have any questions about this or any of the other MCAT content topics, I will try to get out this PowerPoint and recording to you as soon as possible. If if you don't find them soon, just message the group me and just remind me, but I'm pretty sure that I will be able to get it to you by tonight. So if you have any questions, definitely now is a great time for those. And then I'm just gonna stop the YouTube live and recording for now. Let me just